Muscles move you, but how well do you know your muscles? You move because of muscles. You smile right now because of muscles. Usually in your body exams, in your college exams, physiology and anatomy questions on muscles very, very much, prominent. So today I'm going to discuss at least 40 to 50 mind-blowing questions on muscles for us to be able to understand the intricacies of muscles. All right? Because understanding the molecular, cellular, and tissue levels of muscle is extremely important for you, especially for you to get the fundamental characteristics of living organisms and for you to be able to pass your college exams. So as far as you're schooling in the United States or you're schooling in the outskirts of the United States, these questions are extremely important to you for you to prepare for your exams. So I need you to take your pen and paper and follow me as I discuss. So for the first part, I'm going to discuss some little things about the muscles that you need to be aware of. Then I'm going to, ask you some questions. These questions are pointed questions, with the slides for you to be able to get a full grasp of muscle physiology and anatomy. Okay, let's start this way. There are 3 muscle tissues that are important to us. One is the skeletal muscle, another one is the cardiac muscle, and the smooth muscle. They have their differences. The skeletal muscle is very voluntary. That's how I talk. I talk because I want to talk. All right. I move my hand because I want to move my hand. That's voluntary. That's skeletal muscle. Cardiac muscle is involuntary. Right? The heart keep pumping and pumping. You don't control it. Imagine the heart decides to take a break. Do you understand? The same thing with the smooth muscle. Smooth muscle is also involuntary. All right? And smooth muscle are usually muscles that are found in organs in the body. Now, the difference between cardiac muscle and smooth muscle is that cardiac muscle is striated, but smooth muscle is non-striated. So, skeletal muscle, cardiac muscle, both of them are striated. Cardiac muscle, smooth muscle, both of them are involuntary. You need to know this information. Extremely important because it's definitely going to come out in your exam. All right? Now, the next thing we want to discuss is universal muscle characteristics. Very important for you to know. There are major characteristics that you need to know, like responsiveness, conductivity, contractility, extensibility, and elasticity. Very important for you to know that muscles respond to stimuli, of course. They respond to electricity. All right? They can easily be excited. If I touch you, if you get excited, you contract, right? So that leads us to contractibility. The same thing with extensibility and elasticity. I'll try to make this video as short as possible. Now, skeletal muscle is one of the most important aspects that are always coming out in the body exams, all right? So, as I mentioned initially, skeletal muscle subject to conscious control, that's very voluntary, it's striated in terms of the appearance. So you see a lot of Z disc around it, which gives you that striation appearance, all right? Also, there is alternating light dark bands, which I'm going to discuss. Now, skeletal muscle, remember, muscle is an organ, right? Made up of different tissues. Now, if you bring out a cell from those tissues, remember, cells make up tissues. We call it what? Myofiber or muscle fiber. Myofiber or muscle fiber. They are around 30 cm long, all right? And if you bring out a particular myofiber, you will see, peripheral located nuclei around it. That is one of the ways you can know that, oh, this is skeletal muscle. Extremely important for you to know this thing for your exam. Now, when we are talking about connective tissue wrapping, I want you to look at this picture properly. You see, if this is a muscle, the muscle is made up of group of, cells. You see, this is a group of cells. This is a group of cells, right? So what is wrapping this entire group of cells is known as epimysium. Epimysium. Now, the group of cells itself is known as fascicle. If I bring out one of the group of cells, if you're following my mouse pointer, you will see that this group of cells, is being wrapped, this fascicle is being wrapped by perimysium. Now, if among this group of cells, I want to bring out one cell. This is one cell, that's muscle fiber or myofiber that I brought out. This one is now endomysium. All right, endomysium. So there are 3 things you need to know. You need to know what epimysium is, what perimysium is, what endomysium is. I'll repeat. Epimysium surrounds the entire muscle fascicles, right? Now, perimysium surrounds each muscle fascicle. Each is surrounded by perimysium. Now, we have endomysium. Endomysium surrounds each muscle fiber. Remember, muscle fibers are the cells. They make up, group of muscle fibers make up a fascicle. Group of muscle fascicles make up what? The muscle itself. This is your exam question. Now, when you are talking about microscopic anatomy of the skeletal muscle, you talk about sarcolemma, you talk about sarcoplasm, myofibrils, myoglobin, right? It's important for you to know that sarcolemma is the sheet covering is like the plasma membrane of the muscle, rather, right? Plasma membrane of the muscle fiber. All right. So remember, a muscle fiber is a specific cell. We know that the characteristics of the cell. If you don't know these characteristics, I need you to go and look at cell physiology. The characteristics of cell, we talk about the plasma membrane around the cell before you talk about the organelles. The plasma membrane for muscles is sarcolemma. Then when you are talking about organelles, you talk about something like sarcoplasm. That's, sarcoplasm is the cytoplasm of the muscle. So if you enter inside, you talk about cytoplasm of a cell. So that cytoplasm of a cell, for the muscle cell, it's called sarcoplasm. Now, inside the cell, we have something called myofibrils. Myofibrils is like organelles, all right? Like organelles. So it's like organelles, all right? Now, protein bundles for contraction are myofibrils. We're still going to look at it properly. Then we have something we call myoglobin. Myoglobin stores oxygen for muscle activity. All right? Stores oxygen for muscle activity. I'm going to show you all these things one after the other. One of the organelles of the muscle is the sarcoplasmic reticulum, something we know as the endoplasmic reticulum. So there is something we call the T-tubules as well. We call them a triad. So the T-tubules, if you look properly, in between the two greens, you see something dropping down. So I usually call it a T with two wives. A T with two wives, something like this. So, so the end of the sarcoplasmic reticulum is by this side, the other end is by this side, while the T-tubule is in between the two of them. So those two ends known as the cisterns, terminal cisterns, all right? So T-tubule, terminal cisterns, so we call it triad, all right? Very, very important for you to know that the calciums are stored in the terminal cisterns. Calcium are stored in the terminal cisterns. So the T-tubule and the cisterns, all right? Now, if you look at the diagram properly, you will see that this is a thick filament because we have, first, they may ask you in the exam, what the contractile filaments or the contractile proteins. The contractile proteins, the thick and the thin filament. That is, the thick is the myosin, the thin is the actin. All right? So remember that. Those are contractile. They are the ones that are used for contraction. But when you're talking about troponin and tropomyosin, they are not contractile proteins. They are just accessory. Or that are components, all right? Then we have elastic proteins like elastin, like titin. Remember this information, extremely important. This can save your life in the hall. So the elastic proteins, as I said, is the titin, the elastin. Now, there's something else you need to know, which is the dystrophin. Dystrophin is very important because if you look at the diagram, that's why I have to bring out in the diagram here. The diagram, you see the dystrophin. The dystrophin helps to link the actin, that's the thin filaments, the actin, it links it up to the sarcolemma, right? And then to the extracellular matrix. It's very, very important for you to know it because of pathology, all right? Extremely important for you to know it because in pathology, there's a problem with dystrophin, right? Which we will discuss later, not now. This is physiology. Remember, physiology is for you to know the normal so that you'll be able to differentiate it from the abnormal. Now, another thing I want to discuss is the neuromuscular junction. Neuromuscular. Neuro, neuron. Muscular, muscle. Does it make sense? Good. The nervous system and also the muscular system. So if you look at the diagram here of the neuromuscular junction, it's very, very interesting. So this is a neuron. The action potential has already been triggered in the neuron and it comes down here, right? What it does is the action potential does is to activate calcium. So the calcium will enter this synaptic knob. This is a synaptic knob, right? This is a knob, like the extreme end of the nerve, right? This is synaptic knob containing different synaptic vesicles. These are the vesicles. If you look inside, this round thing are the vesicles. What does the vesicle contain? It contains a neurotransmitter. So when this calcium comes, the calcium busts all these vesicles and these vesicles will push out the neurotransmitters through what we call exocytosis into the synaptic what? Cleft. This synaptic cleft, this two area is known as the neuromuscular junction, but this is the synaptic cleft. You see, if you notice, you see the acetylcholine, that's the neurotransmitters falling into the cleft. And immediately they fall into the cleft, what do? All of them go and attach themselves to the receptors. Like in class, I always describe this like, A wants to talk to Mrs. B, so Mr. A looks for a way to pass that information to Mrs. B, because Mr. A cannot talk, Mrs. B cannot talk, and they don't know sign language. So what do? Mr. A writes something on the paper. Oh, I want you to move to the other side. Write it on the paper and throw it down. So what does Mrs. B do? Mrs. B use her hands to pick it up and open the paper using her hands. That her hands she used is known as what? The receptor. So you need a receptor for you to be able to understand what the information in the neuromuscular neurotransmitter is all about. So when the neurotransmitter jumps into, it's just like the information from the nerve to the muscle. But the muscle cannot interpret it except using what? The receptors. So what it does is that it will take the receptors, and through that receptor, it will not understand, oh, I'm supposed to contract. Oh, this is information for me to do to contract. So that's the beginning of the cascade of contraction and relaxation. It's always the beginning. So the information is coming from the nerve. It's just like you have a hot object touch you. That hot object has triggered action potential. The action potential comes to that point where it touched you, right? And that's through the nerve, and gives it information to contract, to withdraw your hand. How does it give the information? Through the neurotransmitter. So it pours the neurotransmitter into the cleft, right? Then the neurotransmitter will match, you understand, the muscle will pick the neurotransmitter up using its hand. That hand is the receptor. Why you need to know about the receptor is because there are some problems that could affect receptors. Like in that DM type 2, as I mentioned initially, in DM type 2 is the problem of the receptor. You are producing insulin enough to mop up sugar, glucose, to convert it to glycogen. You're producing enough insulin. But unfortunately, your system needs to recognize that insulin for the insulin to function. The receptors of the insulin has to recognize it. So when there are no sufficient receptors, what happens? The insulin will be there and it won't be able to function. It will be, it will be meaningless. All right. So very, very important for you to know this information. So for the muscle here, if you notice, it has attached to it. This is the attachment to the receptor. This purple, you see the green attaching to the receptor. So this is the, and this receptor is found in the sarcolemma. If you notice very well, it's found in the sarcolemma. So I've been able to discuss the synaptic cleft, the junctional folds, which contains the ACH receptors. Then we have the Schwann cell, which envelopes and isolates the junction. Then we have the synaptic knob, which contains the vesicles with acetylcholine. These four informations you must know. They are the things that are found in the neuromuscular junction. They are the things that are found in the neuromuscular junction. Extremely important for you to know this information for your exam. Now let's talk about motor units. Sometimes this is one nerve, right? The nerve has dendrites. This is the terminal part of the nerve, just one nerve, right? So this one is innervating one muscle fiber. This another muscle fiber. This another muscle fiber. This another muscle fiber. This another muscle fiber. But it is just one nerve. So that's one nerve with its muscle fibers that innervates is called what? A motor unit. Very, very important for you to remember this. So the definition is that one nerve and all muscle fibers, it innervates. All right. Now, sometimes there are something we call small motor units and large motor units. So small motor units is when one nerve is innervating 3 to 6 muscle fibers. Large motor units is when one nerve is innervating more than 1,000 muscle fibers. All right. Very important. So small motor nerves is used in fine control, like in the eyes and the hands. That's small motor nerves. Very, very precise. But large motor units, they are not precise, but they are used for heavy movements. Extremely important for you to know this. So when we are talking about excitation of a muscle, it always comes from the nerve. You see, the nerve, the action potential comes down here, triggers the breaking of all these things, triggers calcium enters, breaks all these vesicles. They pour their neurotransmitters through exocytosis. They match the in the sarcolemma, and once they match the receptors, what happens? Channels open. Sodium ion opens. Sodium channels open, and potassium channels also open most times. So sodium, we do what? We keep entering inside. Usually the resting membrane potential is around minus 90 MV. So when sodium starts entering inside, it will keep increasing to positivity. Once it passes the threshold, what happens? Action potential occurs. Do you understand? So it's usually sodium enters, potassium goes out. Remember, the major extracellular cation is sodium, and the major intracellular cation is potassium. So usually potassium goes out, but sodium goes in more than potassium, thereby causing positivity in the internal environment that's inside the cell, and immediately causing positivity inside the cell, action potential will be triggered. So the question is, when action potential triggers, what happens? We will get to that, but remember the excitation of muscle. If you look at the diagram properly and the slide, action potential arrives at the neuromuscular junction, causes calcium to enter. Calcium enters, bursts the vesicles. The vesicles pause in the neurotransmitters. The neurotransmitters binds to the receptors. Open the sodium-gated channels. Sodium rushes in, causes negative positivity, which increases the action potential. And immediately action potential is formed, what happens? That's the next question. The action potential will now travel into the T-tubule. So let's go to the next slide and understand properly. Now, if you look at it very well, action potential has been created. You see the arrow? Follow the arrow. The action potential enters the T-tubule. These two areas are what? The sarcoplasmic reticulum end, which is called what we call the cisterns. So this is the T with the two cisterns. This is a triad, right? Action potential enters in this T. And what happens? Immediately enters these calciums that are at the end of this sarcoplasmic reticulum. They are afraid. They run. They start running inwards. And when they run inwards, where did they go to? They run inwards, attaches themselves to the troponin. Oh, troponin, save us. I want you to think the troponin like a door handle. All right? A door handle, you hold the door handle and you open the door, correct? So the troponin is the handle itself. So calcium will come and hold onto the troponin. Once it holds onto the troponin, it will open the door. So that door is signifying what? It's signifying tropomyosin. I told this story in class for the people I've already taught before, that you should think about it like you holding a door handle. You are representing calcium. The door handle is representing what? Troponin. The door itself is representing tropomyosin. So tropomyosin is closing the active site of actin. The wall is the actin. So tropomyosin is closing the active site of actin. So if I use the troponin through the calcium to open the tropomyosin, what happens? The active sites become open. And immediately it becomes open, the myosin, which is the thick filament, will come and patch on the active side, thereby leading to contraction. Mind you, the myosin is able to do that by first of all taking a phosphate and adding it to an ADP to form ATP. Then it will use that energy to match in the active site. Very, very important for you to know this story. Extremely important. I need you to think anatomy like a story. I'll show you some of the questions and you understand what I'm talking about. Now, if you look at it very well, the calcium comes in, matches to the troponin. Troponin does what? Slides the tropomyosin open. Once it's open, what happens? This is the myosin. Myosin comes and laps at the active site of actin and immediately leads to contraction. Extremely important for you to know this. Extremely important. So that's what we call the cross-bridge circle. All right? Very important. So this ATP hydrolysis really makes the myosin to bind in the active site of actin. This is the binding. This is how the ADP takes phosphate to itself. It becomes ATP, and it's now used the energy to do what? To bind. When you are talking about relaxation, you are talking about exactly the same thing. Everything goes backwards. Calcium detaches, but mind you, the myosin still needs ATP to detach. If the myosin does not have ATP, it will not detach. That means contraction will be sustained, and which is exactly what usually happens in people that have died. That's what we call what? Rigor mortis. So relaxation is exactly the same thing backwards. Exactly the same thing backwards. All right? I don't want to go discuss it much so to save our time because we still have a lot of questions to discuss. And this is twitching. All right? Very important. This is twitching. Then there's something we call summation. So there's something we call single twitch. This is single twitch, one stimulus, pium pium. That's it. Then there's something called wave summation. Wave summation is, you see, single twitch, then another twitch, another twitch. This is double summation. 1, 2. Then here, 1, 2, 3, 4. They have to call, a single twitch calls its brothers and sisters, and all of them match together and form what? A wave summation. Now, there's something we call complete tetanus. Complete tetanus is when all these single twitches join together to cause what we call, extended contraction without relaxation. All right? So the contraction continues without relaxation. So I need you guys to be able to understand single twitch, wave summation, and complete tetanus. Now, the types of muscle contraction, we have isometric contraction, we have isotonic concentric, and isotonic eccentric. Isometric. So isometric is tension without length change. There is no length change. All right? Posture is maintained, joint is stabilized. But when you're talking about isotonic, you're talking about concentric and eccentric. In concentric, it shortens while maintaining tension. Like when you are lifting weights or you're pulling actions. We are going to see questions on that. Then we have isotonic eccentric. Eccentric is when it lengthens while maintaining that tension. Like when you are lowering objects or when you have a controlled descent. I'll give you questions for you to be able to understand this better. Now, we talk about muscle energy systems. We have the phosphagen system. We have the glycogen lactic acid system. We have the aerobic respiration. When you need a burst of energy, you're talking about phosphagen system. Immediate energy for 6 to 10 seconds, and it will diminish. Short energy for more than 6 to 10 seconds, maybe like 30 to 40 seconds. Short-term energy, you need glycogen lactic acid. Then when you are, you need a long-time energy, you are talking about aerobic respiration. So these are the three energy systems, because I will ask you this question. So another thing we need to also discuss is oxygen depths. We have oxygen reserves, we have phosphagen restoration, we have lactic acid conversion, we have elevated metabolism, right? So when you do an exercise, usually you're tired, you see him panting, what happens? The oxygen reserves need to be replenished immediately, right? So you have to replenish myoglobin and also the blood immediately, because you expanded a lot of energy when you do those activities. Also, at the same time, you have to review the ATP. You have to review the ATP. And also the phosphate storage. You have to rebuild it. Moreover, you also have to reconvert the lactic acid back to pyruvate in the liver. The lactic acid is what the body produces during aerobic respiration, and that is what gets you fatigued and tired during the joints when you're running. All right? So it has to go back, reconverted. Also, elevated metabolism, helps to support increased post-exercise rates, because when you finish exercise, you are breathing, you are tired, right? Your body is still working. So there is still elevated metabolism. So these things are what we call oxygen depth. You have to know oxygen reserves, phosphagen restoration, lactic acid conversion, and elevated metabolism. Extremely important. So as I was saying initially, when you are fatigued, it's as a result of you have depleted the ATP. Your ATP has finished. Lactic acid accumulation lowers the pH, as I mentioned initially. Then your potassium is imbalanced. Do you understand? And even mentally, your CNS systems are limited. At that time, everything was concentrated. All the energy you have in your body was concentrated on the muscles. So everything is working hyper. So you become so fatigued and tired, as you can see these athletes right now. There is also something we call slow twitch, red muscles, fibers, and fast twitch, white muscle fibers. So we have red muscle and white muscle. These are muscle fiber types. You have to know this. What are the characteristics? Characteristics of slow twitch, that's red muscle. It is slow, that's type 1. Fast glycolytic is type 2. It's very, very fast. The color, the red ones are always red because they have a lot of myoglobin, a lot of oxygen. But the fast twitch are pale. They have less myoglobin. Mitochondria is abundant in the slow. Mitochondria is few in the fast. Fatigue resistance is very, very high in the slow. Fatigue resistance is very, very low in the high. Twitch duration, 100 ms. Twitch duration, 7.5. So the fast is very fast. It's twitching fast. Pew, 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 pew. But this one twitches slow. Twitch. All right? Now, the primary use, the slow, is for posture and endurance. That's why it's red. Why the fast is for quick movements. You see how my hand is moving? I'm using my fast twitch. I'm using my type 2. All right? My type 2. That's fast twitch white muscle fiber. That's what I'm using. All right? So very important. But if I want to lift an object up, I'm using the type 1. That is slow oxidative, or what we call slow twitch red muscle fibers. I need you guys to understand the difference between fast and the slow. I'll give you a question that will, you will see the differences. So how do you strengthen your muscle? You are focusing on muscle size, fascicle arrangement, and motor units. Like you see some people lifting weights. What are they doing? They're increasing their muscle size, the size. Remember I told you something about hypertrophy. Hypertrophy is when the size of muscle is increasing. I was telling you the difference between hypertrophy and hyperplasia. Hyperplasia is usually increasing number, quantity, but hypertrophy is increasing size. So usually it can be muscle size, fascicle arrangements, and motor unit recruitments, right? So some people do resistance training and endurance exercise. It depends on what you want. Resistance training, hypertrophy, they stimulate hypertrophy, increases myofilaments. Endurance training improves fatigue resistance, increases mitochondria to endure. There are some noteworthy muscle disorders. Some noteworthy muscle disorders like the muscular dystrophy, which is as a result of mutation in dystrophin. I mentioned it earlier. Dystrophin links actin to the sarcomere. Then we have the myasthenia gravis, which is an autoimmune deficiency that affects ACH behavior. We also have tetanus, which is spastic, which leads to spastic paralysis, and we have botulism that leads to flaccid paralysis. There are some people that put Botox here so that they will not have, so that they will not have wrinkles. All right. So that is botulism. For muscular dystrophy, we discussed that it's hereditary disease causing muscle degeneration as a result of the dystrophin having a problem. That's connection. You understand? Having a problem. The most common type is the dystrophin, muscular dystrophy. That's the most common type, which I told you is the dystrophin mutation, right? It usually affects the skeletal, the cardiac, the respiratory muscles, and also it's gradually, before you know it, it increases. And there's something we call the grower sign. If you look at how this patient is getting up, that is usually, the grower sign is usually a sign that shows, oh, this is definitely muscular dystrophy. It can be Duchenne most times because that's the most common. Then we have myasthenia gravis, which is usually targeting the ACH receptors, just like type 2 diabetes, the same thing, right? So those receptors are being targeted. So it's not about the transmitters. There is a lot of transmitters there, but the problem is the receptors. So transmitters are produced, but there's no receptors for them to anchor on, So like the illustration I gave, imagine writing something, Mr. A writes something on paper and throws it to Mr. B, Mrs. B, but Mrs. B does not have any hand or anything to pick it up. So she will never know what is inside. Make sense? Symptoms are usually muscle weakness, especially facial, and treatments, we usually give cholinesterase inhibitors, right? Because cholinesterase usually break down acetylcholine. So we are trying to, leave acetylcholine to stay there much more. So we inhibit cholinesterase enzyme. Now, tetanus and botulism. Tetanus causes something we call lockjaw, right? It's, it also causes opisthotonus, which I'll show you probably. Now, both of them are just, tetanus has toxin. That toxin blocks the inhibitory signals. Now, if the inhibition, the signals to, stop the contraction is blocked, what will happen? The contraction will continue. That's what we call paralysis. But this paralysis is contracted paralysis. That is spastic paralysis. So you see rigid contractions, you understand? It always starts in the jaw. That's what we call lockjaw. Now, but botulism is also paralysis, but this time around is different. It's not rigid. Instead, it's flaccid. Flaccid. Why is it flaccid? Remember, there must be relaxation. There must be relaxation, and there must be contraction. Now, botulism, what it does is that toxin will go and block AC release, right? So the vesicles are not able to release ACH, acetylcholine. So if the neurotransmitters are not able to, you cannot write on paper and send to Mrs. B. So how will the information be transmitted? No way. So there will be no contraction. It becomes flaccid. So that's flaccidity, inability to contract. Sometimes it affects breathing muscles, and the person muscles will collapse and the person dies. That's why when you are even doing Botox application, you have to be extremely careful. It must be handled by professionals. So one of the questions that they'll be asking you is about the facial characteristics. For example, they may tell you all muscle types will respond to an electrical stimulus because all muscle cells are dash. Why do they respond to electrical stimulus? Because they are excitable. All right? Because they are excitable. Because you see options like, oh, because they can contract, because they are striated, because they are involuntary. No, but because they are what? Excitable. Also, you'll be asked questions on questions on skeletal muscle. Like, is it voluntary? Is it striated? Does it has multiple nuclei? For example, I can tell you skeletal muscle is called dash because it's usually subject to conscious control. It's called what? Voluntary. Conscious. I told you skeletal muscles are voluntary, but cardiac and what? And smooth are what? Involuntary. So conscious control, skeletal, voluntary. Very important. So remember these characteristics is always very important. The responsiveness, that's excitability, conductivity, contractility, all right? Even extensibility. So we have excitability, extensibility, contractility, and what? Conductivity. Do you understand? This, because they may give you this and they will put one and tell you which one is not. Mind you, always read the question before you answer. Which one is not? You should know which ones are and know which ones are not. So you must learn this because a lot of questions will be coming out based on muscle type characteristics. Also, I need you to know the difference between the collagen and muscle, right? So collagen is very, very different from muscle. It does not have the characteristics of muscles apart from elasticity, right? So collagens are elastic. In short, it is the collagen in the muscle that helps it to expand. So if they're asking you which of the following is a property of collagen, please, the answer is always elasticity. It is not contractility. It's different. Elasticity is different from contractility. It's different from excitability. It's different from expandability. It's different from impermeability. It's always what? Elasticity, mind you. And it is this collagen, that usually help, as I mentioned initially, helps skeletal muscles when they lengthen. It helps them to resist excessive stretching, subsequent injury to the muscles. All right. So when I stretch my skin now, for it not to injure, it is the collagen that will help it to, resist that extensibility. All right. So I need you to have that in mind. Very, very important. And I told you about fascicles, correct? So I told you that if you look at this diagram, what are you seeing? You will see this is muscle fiber. You see a bundle of muscle fiber. You see different muscle fiber bundles. Each of these bundle is called what? Fascicle. Fascicle. So if I ask you a muscle of muscle fiber is known as what? Fiber is known as what? It is what? Fascicle. Don't forget. What is wrapping up fascicle? It is what? The perimysium. What is wrapping up muscle fiber? It is what? The endomysium. What is wrapping up the entire fascicles? It is what? The epimysium. Always very important for you to know. Now, what do you know about the sarcoplasmic reticulum? Because I will ask you a question on sarcoplasmic reticulum, for example, or you may be asked a question if it's your board, and the question will be targeting to know if you know that sarcoplasmic reticulum is what houses, they are the two cisterns, like that's the two sisters, right? They are what houses what? Calcium. So when the muscle cell is excited, when action potential runs in straight forward, what happens? The cell becomes excited. It pushes calcium out, releases calcium. So if they're asking you about it, because they may ask you what is true or about sarcoplasmic reticulum or what is false about sarcoplasmic reticulum, you should know that sarcoplasmic reticulum releases calcium when a muscle fiber is excited. This is very important. You understand? It doesn't synthesize the ATP. It doesn't do anything with sodium. It doesn't do anything with acetylcholine. It is specifically calcium release when the muscle is excited. You must not forget this. All right? Now, they may ask you something similar to it. So I need you to, when you know these concepts, what I'm teaching you is concepts. When you know these concepts, it will help you to be able to understand any question outside these concepts. Now, this is a place that you'll be tested, and I really need you to get the concepts. If you look at the diagram, all right, this is a Z disc. This is a Z disc. They may ask you, what is attaching to this Z disc? Or rather, they may tell you, this particular space, this place, you see this place is thick, this place is thick, this place is thin. They may ask you, what is this thin part? this light part. This part is called what? The I band. If you see my mouse, this is the I band, right? This thick place, that's from here to here, is called what? It's called the A band. That's dark band. Dark band, light band. Dark band, light band. Very, very important for you to know. Extremely important for you to know. All right? Why the sarcomere is here, that is from this Z disc, another Z disc, is known as what? The sarcomere. It's known as the sarcomere. Now, if you come to this dark band, there is a light part in between it. There's a light part in between it. This light part in between is known as what? The H zone or the H band. All right? The H zone or the H band. In the middle of this H zone, there's something we call the line. You will see this very well if you come over to this part. You see, this is the H zone. In the middle is the line. So I need you to know the Z disc, the sarcomere, what the sarcomere is, what the H band is, you know, what the I band is, what the A band is, what the line is, what the H zone is. You really need to know this. All right? Now, the elastic filaments also attaches themselves, you know, to the Z disc, as well as what? The thin filament. So this is the thin filament. This is the thin filament. Why this is the elastic filament? And this is the thick filament. So the thick filament, that is the myosin. The thin filament, that is the actin. You need to know, study this. Take your time. Study this diagram. Very important for you to know it. Because they may ask you some questions. Like, they may tell you which of the following contains overlapping thick and thin filaments. That overlap is known as what? A band. Very, very important. That overlap is known as what? A band. This is the overlap here. It's known as A band. But you see, so the actin is from here to here. Another actin from this Z disc is from here to here. So this space where there is no actin is known as what? It's known as what? It's known as what? Put the option, answer in the... I want you to pause this video and put the answer in each of them. So I will be asking the question and you tell me. So actually, that place is, you know, the line in between. The line is in between. The H zone is, you know, this area, these two areas where there is no actin. See the actin ends here. That's the thin filament ends here. The thin filament ends here. So this space is known as the H zone or the H band, as I was asking initially. I need you to pause it and tell me what is A band. Pause it, tell me what is the I band. Pause it, tell me what is line. Pause it, tell me what is the H zone. Pause it, tell me the part that overlaps the actin and the myosin is what? The distance between the two Z discs is called what? I need you to pause it and tell me the answer. Put it in the comment box, all right? Also, they may ask you what striation is, right? You tell them that striation is that describes the alternating light and dark bands. The alternating light and dark bands separated by the Z disc, that is striations. It's important for you to remember this. Also, they may ask you about the troponin. What did I say is the troponin? The door handle. So what holds the door handle or what accepts the door handle? That is calcium. So they may ask you a question that the protein that acts as a calcium receptor in skeletal muscle is what? It is what? Troponin. You can't forget this. You really can't. They can also ask you about the tropomyosin, which closes the active site of the actin. Another question they can ask you is dystrophin. Dystrophin, they may ask you, you know, what is defective in the muscular dystrophy? It is dystrophin, and you need to know what the dystrophin is or what it attaches. So usually, normally, the dystrophin, you know, it's between the outermost myofilaments and the sarcolemma of a muscle fiber. So usually, the actin and the sarcolemma, actin and sarcolemma, actin and sarcolemma, or the outermost myofilament and sarcolemma. Very important for you to know this. Extremely important. They may ask it in any form. They may ask it in any form. They ask you, what is dystrophin? They may ask you, what is muscular dystrophy? They may ask you, what does dystrophin connect? And that is when you tell them the outermost myofilament, that is the actin, and also the sarcolemma. And also, sometimes they may ask you how many somatic neurons stimulate one muscle fiber. It is one. It is one. One somatic motor neuron stimulates one muscle fiber. It's never two. Now, they can ask you about recruitments. Can you remember what recruitment is? Pause the video, put it in the comment section, or pause it and just try it. All right? Very, very important. So recruitment is usually the process of engaging more motor units to increase the strength of a muscle contraction. right? You want to increase more motor units. So I want to push this chair with 1 motor unit. Oh, I cannot. What do? I increase my power. What is that increase in power? Recruitment. You are calling everybody. It's different from wave summation. Different from wave formation. All right? Different from complete or incomplete tetanus. Different from tetanus. Recruitment is, please come and help me. Oh, hey, muscle, motor units, come and help me. Motor units, come and help me. Before you know it, there are a lot of motor units and they do that activity. Very important for you to remember. So the process of engaging more motor units to increase the strength of a muscle contraction is recruitment. I've discussed twitch, I've discussed summation, I've discussed tetanus. So for example, if maybe you want to lift a box and someone places an extra weight on top of the box, what do? You have to do what? Contract more to lift that box. That is what recruitment is. So you are recruiting more muscle fibers. So if I ask you a question like that, for example, I ask you a question that, oh, as you are lifting a weight, a box, and somebody puts extra load on it, you have to contract more. What is it called? It is called recruitment of more muscle fibers. Now, do you remember how excitation works? What did I tell you about stimulating muscle contraction? I told you that, first of all, the acetylcholine is released from where? From the synaptic vesicles that are found in the synaptic knob. What causes that release? Calcium. How is it released? Exocytosis. You have to pay attention. You have to know what they're asking, because this question can be thrown to you from different angles. But the most important, remember that synaptic vesicles does what? Houses acetylcholine. That is what releases acetylcholine. How is it released? It's stimulated by what? Calcium. What causes calcium stimulation? Action potential from the nerve. When calcium stimulates the vesicles, how does it release this? It pours it out. Exocytosis. Pours it outside the cell. So when I ask you this, you should remember what synaptic vesicle is. Also, you need to know what the neuromuscular junction of a skeletal muscle is, because this is the place, this is the terminal. So this is the axon terminal, you see? This is the muscle fiber. So in between these axons, so this area is known as what? Neuromuscular junction. Between them is this cleft, you understand? So if I describe this for you, you should be able to tell me that this is, neuromuscular junction of a skeletal muscle. Extremely important. So I need you to also know the sequence, especially what happens in the neuromuscular junction. When acetylcholine comes in, what does it do? It stimulates the receptors in the neuromuscular junction, correct? When it stimulates these receptors, what happens? I want you to put the comments write something in the comment section before I give the answer. Okay, so what really happens is that it will lead to opening of sodium-gated channels, right? Thereby increasing the permeability of sarcolemma towards sodium. So sodium will start influxing, increasing. As sodium is increasing, it increases the positivity, that's the threshold. When it gets to the threshold, action potential. Then the action potential will now enter the T-tubule, push the calcium inwards. Calcium matches to the troponin. Troponin opens the tropomyosin opens the active site of actin, and tropomyosin, with the aid of ATP, laps on it, leads to contraction. So everything works with permeability of the sarcolemma to what? Sodium increase. And that is as a result of acetylcholine stimulating the neuromuscular what? Stimulating the receptors in the sarcolemma. Make sense? You can't forget. Also, another thing you need to know is the resting membrane potential, right? The resting membrane potential is actually the intracellular environment, the negativity of the intracellular environment. That's why here I wrote the resting membrane potential minus 90 MV is the intracellular negative charge. So the intracellular environment is negatively what? Charged. Anytime you see RMP, resting membrane potential, know that they are asking you about the charge of the intracellular environment, which is always what? Negatively charged. You can't fail this in your exam. You can't afford to fail it. Yes, did you remember what I told you about tetanus? I told you that tetanus is what? Is lack of inhibitory signals. So if I tell you that, oh, something happened though, and there was an absence or inhibition of acetylcholinesterase at the synapse, what will it lead to? It will lead to tetanus. All right? Bear that in mind. That's the difference between tetanus and what? Botulism. And also, I may ask you a question on myasthenia gravis, which I may not ask, but for your board exams and for other college exams, some professors might want to test your myasthenia gravis. You should know something very important, that myasthenia gravis is autoimmune attack and is usually targeting the ACH receptors, and its symptoms is usually muscle weakness, muscle weakness, especially facial. All right. And the drugs that treats myasthenia gravis is usually targeting what? Is trying to inhibit the function of cholinesterase. Bear that in mind. And also, when we are talking about the sarcoplasmic reticulum, having a lot of calcium storage, we are talking about skeletal muscle. So if I ask you which muscle type depends solely on the sarcoplasmic reticulum as its major calcium source, you should be able to tell me that it is what? Skeletal muscle. All right? So I may ask you about muscle contraction. Very, very important that a lot of colleges ask this question. They may be asking you about, what uncovers the active sites of actin. You should know that it's tropomyosin. I've described it a lot. It's just the door. Tropomyosin is that door. The handle is troponin. So the door is covering the active sites. Immediately the calcium perches on the handle, calcium squeezes the handle, and the handle turns the tropomyosin. So if you notice, this is the tropomyosin. You see that long door covering all the active sites of actin. That's the tropomyosin. Very, very important for you to know. So immediately it opens and the myosin binds on it. It leads towards, there is a binding, all right, of the myosin heads with the actin filaments. Very important for you to know because they will actually cross bridge forms what? What is even cross bridging? So cross bridge between the binding sites on actin and what? And myosin head. Actin myofilaments and what? Myosin head. Please remember this. And if they ask you what propagates the action potential, it is sarcolemma. Action potential is propagated along the sarcolemma. It's very, very important. All right. When they are trying to test you on action potential, they may tell you that increased calcium ion permeability of the presynaptic terminal cell membrane is caused by what? Action potential, of course. Right? Of course. So I told you that, if you remember vividly, I told you that, here when action potential from the nerve comes, it will trigger calcium to enter inside and break up these vesicles and burst them into the clefts. Always remember this question because it's always on your college exams and it's always on your boards. And also, I want you to know that the minimum stimulus needed to cause muscle contraction is called threshold. It is called what? Threshold. And a skeletal muscle generates the greatest tension when it is partially stretched before being stimulated. Because they can tell you when it is greatly stretched before being stimulated. That is not the answer. When it is fully relaxed, no. When it is well rested, no. When it's high in lactate concentration, that is even when it gets weaker. All right. So you should be looking for, it is partially stimulated before being, partially stretched before being stimulated. That must be the answer. All right. Just like when you warm your car, it's partially on, you've not started moving. It makes sense than when you just on it and jump into it and start moving immediately. All right. So the greatest skeletal muscle generates the greatest tension when it's partially stretched. And it must get to a threshold. Minimum stimulus need to get to threshold before muscle contraction happens. If it doesn't get to threshold, contraction will never happen. That's what we call all or none law. And if you look at this diagram I told you about complete tetanus, it's very important for you to know because they can ask you that if one nerve stimulus arrives at a muscle fiber so soon that the fiber does not relax at all from the previous switch, right? What will happen? It is complete tetanus, right? If you notice, this comes before it's relaxing, this one comes, 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 and all of them causes complete tetanus. Unlike this one that will go and drops, go and drops, and continue going. Do you understand? Also, I will ask you about isometric and isotonic contraction. A question like shortening a muscle while it maintains constant tension is called dash. That's what we call constant tension. Constant tension. That was what we call isotonic contraction. Now we are trying to differentiate between isometric and isotonic. Isometric and isotonic. All right? So bear in mind that a shortening a muscle while it maintains its constant tension is isotonic. But for isometric, there is tension without changing length. But the other one, there was change in length because we shortened it. Change in length, you shortened. All right? Then before you start talking about concentric and eccentric. All right? Whether it is shortening or lengthening, but there is a change in what? Length. If there's no change in length, that is what? Isometric. So you have to bear in mind, because I can ask you, which one is true about isotonic eccentric contraction or isotonic concentric? So when I'm asking isotonic eccentric, I'm telling you, you have to tell me that the muscle lengthens, but tension remains what? Constant. Lengthens, but tension remains what? Constant. That's eccentric. But when I'm asking concentric, tell me that the muscle what? Shortens, and why the tension remains constant. It's very important for you to know this. Also, you need to learn about the creatine kinase. I didn't mention it, but the creatine kinase helps to transfer phosphate from the CP to ADP. All right, that transfer of phosphate is catalyzed by the creatine phosphates, or the creatine kinase, not phosphate, creatine kinase. All right, very, very important. Then you need to know about the phosphagen system, immediate energy, glycogen, lactic acid, short-term energy, aerobic respiration, long-term energy. And as I mentioned, remember this, all very, very important. Phosphagen restoration, rebuild ATP and CP stores. Lactic acid conversion converts back lactic acid to pyruvate. Where? In the liver. So it means that 80% of the lactate produced by the skeletal muscle is converted to pyruvate rather, by the what? Liver. By the what? Liver. So if I ask this question, you should be able to get it. If any of your professors in your colleges ask this question, you cannot afford to fail it. And you may be asked a question about, why does one continue to breathe heavily after rigorous activity has stopped? Maybe you did a treadmill, like 200 at a go. And when you stop, you see yourself panting. Why? It is because the body requires more oxygen to restore levels of ATP and creatine phosphate, CP and ATP. Very, very important. They have to be replenished. If you notice what I mentioned initially, they have to be replenished. Very important. And also you can be asked about atrophy. You need to know what atrophy is. Atrophy is the loss of muscle mass from lack of activity. Hypertrophy is increase in muscle mass. All right? Extremely important for you to know this question. And also you need to know about fast twitch. You need to know about slow twitch. And the cardiac muscle has the highest mitochondria, the largest, the cardiac muscle. So if I ask you which one of the following has the largest mitochondria, it is the cardiac muscle. It is the cardiac muscle. Think about it. The skeletal muscle doesn't have the highest mitochondria. Some people will say, oh, but we do the skeletal muscle every yes. But sometimes when you are resting, the heart is still beating. It needs that constant supply of energy. So that's why it has a lot of mitochondria. Like if you reason it, you see that you can't fail that if you meet it in a body exam. Also, remember I told you that resistant training stimulates what? Hypertrophy. Resistant training, hypertrophy. Resistant training, hypertrophy. But endurance training is fatigue, to improve fatigue and to increase mitochondria. Very important. Which muscles can contract without the need for nervous stimulation? Skeletal muscle must be stimulated for it to contract, but smooth muscle and cardiac muscle does not need. That's why they are involuntary muscles. You understand? They don't have a need for it. Bear that in mind. All right? So you can be given a muscle fiber like this, and you'll be asked to identify some of the structures. All right? Very important. You may be asked to identify the sarcolemma. You may be asked to identify the triad. All right? You may be asked to identify the myofibrils. Or you may be pointed to some of these points and you're asked to identify which one is the I band, A band, line, H zone. All these things is important for you to study. So this is where we come to the end of study for today. We've discussed more than 60 questions, and I wasn't naming the questions, I wasn't numbering them, but I need you to. These are not all-inclusive lists, especially for you going for your board exams. I want you to take time to find out more questions. All right? You can take this topic, put it in an AI, and tell an AI to generate 100 questions for you. All right? Generate these questions. The more questions you attempt, the better for you. Look for question banks and answer most of these questions. By so doing, you'll be well prepared for your exam. Thank you so much. If you have any comments, please put it down. If there's a way I can make this video better, put it down in the comment section. If you want me to treat a certain topic that you are finding difficult for your college exam or for your board exams, put it in the comment section, and I will just discuss questions. Bear in mind, this does not substitute for your slide. Please do well to read your lecture slides, your lecture notes. Use this just something you need to do a review on. So this is like a fast review, giving you questions for you to think about. Look at those questions. Look at things that they could deceive you. Similar questions. So by so doing, you'll be well prepared for your exam. I'll see you in my next video. I'll be talking about muscle itself. This time around, no longer muscle physiology, muscle itself. And we'll be discussing about 40 to 50 questions on normal muscles.